Thank you so much for the introduction, and I'm super excited to be here today to talk to you all about the work that I've been doing on the prototypic crAssphage. So first, just to kind of zoom out a little bit, our lab is very interested in studying the gut microbiome, which is the collection of all the bacteria, viruses, and microeukaryotes that reside within your gut. And we actually know a lot about the bacteria that are in your gut and all of the things that they do for your body every day, like producing vitamins, helping you break down your food, modulating your immune system, and providing resistance to pathogen invasion. But we don't understand the other components of the gut microbiome all that well. And specifically, I'm going to talk to you today about gut bacteriophages and how we have very little understanding of their biology as a whole. And so largely we think that gut phages actually might not be very lytic. And so they're not following these kind of classic lifestyles that we think of when we think of bacteriophages, where they replicate a ton and kill their host. And this also makes them much more difficult to culture and therefore much more difficult to understand their lifestyles. So the phage I'm going to talk to you about today is called the prototypic crAssphage or Carjivirus communis. And this phage is really interesting to us because it's incredibly prevalent and incredibly abundant. And so we think this phage is present in tons of healthy individuals throughout the world, and that it is found in up to 70% of all people in Western populations and can make up to 40% of the gut virome globally. This phage was discovered in 2014 through computational methods, and it was thought to have this circular 97 kilobase double-stranded DNA genome, but it was really difficult to predict what its bacterial host was. And despite a lot of attempts by different labs, no one was able to culture this phage. And so it's been really hard to uncover its biology. And so when we know that it's present in tons of people, we actually don't really know what it's doing within the gut. But we do know that it's not following these conventional lifestyles that we tend to think of when we think of gut bacteriophages or bacteriophages as a whole. And so there's no sequencing evidence that we can find that this phage is integrating into bacterial chromosomes. And while it seems to encode all of the genes that it needs in order to undergo a lytic replication cycle and burst out of a host cell, we and other labs have been unable to get this phage to form a plaque on a plate, which means that it's not strongly lytic if it is undergoing this lytic replication cycle. And this also makes it really difficult to culture because traditionally you culture phages by picking these plaques off of a plate. And so looking at some genomics and trying to understand this phage, I hypothesized that C. communis might use a phage-plasmid lifestyle for extrachromosomal maintenance rather than integrating into its host genome. And so phage-plasmids are essentially just these elements that can replicate both as a phage and as a plasmid. And we typically characterize them by looking in phage genomes for plasmid-originating genes and plasmid genomes for phage-originating genes. And it's thought that actually 5 to 7% of all phage and plasmid genomes that are available in public databases are actually phage plasmids. And so I don't have time to show you any of the data today, but I'm just going to present you the model for how we think this phage is working and what its lifestyle is. So I've been able to show that this phage can actually replicate on a wide variety of hosts. So typically we think of phages as having a very narrow host range and being species or even strain specific. But this phage seems to be able to replicate on 3 different species of bacteria and different strains of these bacteria, including Phocaeicola dorei, Phocaeicola vulgatus, and Bacteroides stercoris. As I mentioned, we don't see any integration of this phage, and we don't see any plaque formation by this phage. So instead, we think that this phage was largely maintained intracellularly at roughly 10 copies per cell in actually a linear form, with these end protection proteins bound to the linear ends of the DNA so that it doesn't get broken down by the cell. We think that it also has 2 origins of replication, and that likely it can fire from one or the other depending on which lifestyle it's using, phage or plasmid. We think that this phage can sometimes exist circularly, although we think the dominant version of the genome is this linear form. And we also think that this phage is capable of mobilizing through a MOB-C protein through existing conjugative structures into neighboring cells. So this allows it to move between cells without the need for actually forming phage particles. And this might be one way that this phage has evolved to not need to be super lytic and still be able to spread through bacterial populations. We do see some cases where we think this phage is undergoing lytic replication. And when it does undergo lytic replication, we think that it probably releases somewhere around 200 particles per lysis event. And we're actually able to visualize these particles through both TEM and cryo-EM. And so I just like to thank everybody that has made all of this work possible, especially Emmy and Gavin for letting me do this crazy phage project when neither of them were working on phages when I joined the lab. And Angela and Yaka, my co-authors, for really helping me with a lot of these experiments and computational analyses.